All right, welcome back. In the second video for Chapter 12, we're going to look at test crosses and the multiplication and addition rules. All right, if we look at Mendel's first law, we learn that heredity is particulate. Remember, we inherit alleles or different versions of a gene, such as big P or small p when we're talking about flower color. We saw that for true breeding parents, like in the pea plants, they carry 2 hereditary particles for each trait, such as big P, that's 2 of them, or small p when we were looking at the white flowers. Remember that these alleles, he calls them particles at this point, come apart during gamete formation. So remember, parents are diploid, and then the gametes, however, are haploid, the sperm and egg that are formed. And only one or the other particle, so only one of these alleles, is passed on through the gametes, and they recombine randomly. So the sperm and egg, for example, recombine randomly during fertilization to produce offspring. So if I had a heterozygous individual, for example, crossed with another heterozygous individual, the gametes could be big P or they could be small p, or this one could have a big P or a small p. And when they recombine, you could have them come together randomly. Big P could be combined with big P, or big P could be combined with small p, or this small p could go with that big P, or this small p could go with that small p. And that's that random recombination we see. We're going to see a test cross in the next slide, and that will allow us to tell whether or not something that has a dominant phenotype, such as purple flower color, whether that is actually a homozygous dominant individual or a heterozygous individual. So whenever you're not sure what the parent is, if it's true breeding or a monohybrid organism, then what we can do is perform a test cross with a homozygous recessive organism. Let's look at the test cross from our book. So we have true breeding parents, big P, crossed with small p, and this would be a hybrid individual, heterozygous individual. The test cross is shown here in the F1 generation with, this is the tester. So a tester is always a homozygous recessive individual. And if this is my unknown individual, if I didn't know the actual genotype of that individual, I could make a prediction based on the results I get in the F2 generation. So here, what would happen would be, I would get either, let's see, big P with small p, or big P with this small p, or small p with small p, and small p with small p. So looking at those possible combinations, I predict that I would get a 50-50 ratio or a 1-to-1 ratio of purple flowers to white flowers. Punnett squares are great to use when you're looking at one characteristic with 2 traits, such as flower color, purple or white, and you want to look at the genotypic and phenotypic ratios of their offspring. So how do I use a Punnett square? Let's say I have a homozygous purple flower plant and a homozygous recessive white flower plant. What you do on the edges, as they've done here, is separate the gametes. So I know that this is my organism, it's diploid. Gametes are haploid. So I know one of the big P's would go here, the other gamete would go here. And you do the same thing with the other parent. So for the other parent, they put their gametes here, small p. Remember, gametes are always haploid. And to see the offspring are what are inside the boxes. That's the F1 generation. If out here we have the parental gametes. So when you put the gametes together to regenerate the diploid organism, these are the potential offspring you will see. So you can see from the true breeding parents, all of the F1 generation, this is 100% big P, small p. So 100% of the genotype would be big P, small p, 100% of the plants would be purple. If I have a monohybrid cross this time, again, you separate the gametes for one parent on one side of the Punnett square. You do the same thing for the other side of the Punnett square, and then you fill in the genotype in the small boxes. That would give you the next generation. Oops, this is small p, small p. So I can see here I would have a 1 to 2 to 1, 1 to 2 to 1 genotypic ratio, but a 3 to 1 phenotypic ratio, 3 to 1 purple to white flowers. So now let's put the Punnett square together with the test cross. Remember, the purpose of the test cross is to determine whether an organism that's expressing the dominant trait is homozygous or heterozygous. Is it, for example, in this case, we're looking at the seed color of the pea plants. If I see a yellow seed, is this big Y or big Y small y? Is it homozygous dominant or is it a heterozygous individual? Whereas the recessive individual is homozygous recessive. So the tester is always homozygous recessive. Based on the offspring I get, I can predict whether my unknown individual is homozygous or heterozygous. So let's put the gametes for the tester on one side, like they did here, small y, and for the gametes of the unknown parent, I'll put them on the other side, big Y question mark. I know it has at least one big Y because the seed is yellow. And if I get 100% of these F1, if this generation, if 100% of the offspring are all yellow seeds, and I have like hundreds or thousands of seeds, then I am pretty sure that this parent is big Y, big Y. But if I have, again, if this is my unknown individual, and this is my gametes for my tester, if I see any green seeds, then I know that for sure that the parent was big Y, small y, that it was heterozygous, because there is no way to get green seeds in your offspring unless the second allele was a small y. So if you see a 50-50 or a 50% 50%, which is also a 1-to-1 ratio of yellow to green seeds, then the original unknown parent must have been heterozygous. So this is extremely helpful if you, again, don't know if a parent is homozygous dominant or if it's heterozygous for that characteristic. Our textbook has a great question from the reading section 12.2, and the question is right under the figure 12.5, the test cross we just saw on the previous slide. It says in pea plants, round peas are dominant to wrinkled, and we have a test cross between a pea plant with wrinkled peas, so that must be my tester, and some plant of unknown genotype that has round, so round question mark with my tester. I end up with 3, so my F1 generation only has 3 plants. All of them are round. So it must be like that, right? Because I know it has to get a small r from the tester, and to be round you have to have at least one big R from this parent. From this data, can I tell if the round pea plant is homozygous dominant or heterozygous? I mean, it is 100% round, and earlier we said this must mean that that's homozygous dominant. But in this case, I can't be sure because that's too small of a sample size. So I still don't know. I would say I'm not certain. I'm not certain. If the round pea plant is heterozygous, if this was heterozygous, big R, small r, what's the probability that a random sample of 3 progeny peas will all be round? Okay, let's see. If this was big R, and this is small r, what's the chance that I would get that? So I could do my Punnett square, big R, small r, small r, small r. So it looks like half a chance every time. So it would be 1/2 times 1/2 times 1/2. That would be 1 out of 8. So I have 1/8 chance that all 3 progeny peas will be round. All right, again, Punnett squares are really great at predicting outcomes for our offspring. If you have parents that are homozygous, you really only need a 1-by-1 box because this one can only donate a big Y and this one can only donate a small y. So 100% of the offspring will be hybrid, will be heterozygous. But now these offspring can generate 2 gametes. So it's nice to have a 2-by-2 box, 2-by-2 box. And when I look at my F2 generation, I again see that 1 to 2 to 1 genotypic ratio. 1 big Y, 2 big Y, small y's, and 1 small y, and a 3 to 1 phenotypic ratio. Again, these are the ratios that I asked you to memorize for a monohybrid cross. If you ever see the ratios without knowing what the F1 generation is, you can predict based on this. There's a lot of probability when we get into genetics and predicting our offspring. So these are chance events, and if something has a 0 chance, that means it will not happen. So there's no chance. The opposite of this would be a probability of 1, which means it will for sure happen. It's 100% likely to happen, so there's no chance here either. Anything that's between 0 to 1 is a chance of happening, such as flipping a coin. There's a 50% chance that heads will be facing up. There are 2 basic rules we follow when we calculate probability for genetics problems. Whenever you see the word and, when 2 things have to be true in a certain order, we multiply. If you see the word or, then we're going to add. So here's an example from our book. Have you guys seen dice before? So singular is die, so I'm looking at one of these dice. If I want to roll a 5, I know there are 6 sides, and 5 only shows up once out of the 6 times. So the chance of rolling a 5 is 1 over 6. If I want to roll a 4, the chance is also 1 over 6. So if I want to calculate the chance of rolling a 5 on the first die and a 4, this doesn't say 5. I couldn't find a picture with 5 on it. Pretend that's 5. And then a 4 on the second one. Then I see the word and. I want both to be true, so I multiply the 2 probabilities and I get 1 out of 36. Let's try a question that's not in our book. Let's say a couple is trying to have children and they want to have 2 females. What's the probability of having 2 that are both female? So the probability of having a first one is half, right? You can get a male or female. And, so if I say the word and, I'm going to multiply. The second child will also be female. Then my total probability, my chance is 1 out of 4 or 25%. If there are multiple ways for an outcome to occur, we usually see the word or instead of and in this case, we use the addition rule. So let's say that someone can win if you roll a 4 or a 5 on one of these dice. So the chance of rolling a 4 we saw earlier is 1 out of 6, or the chance of rolling a 5 is also 1 out of 6. So if you see the word or, because you could win with 4 or a 5, then we add. So 1 out of 6 plus 1 out of 6 is 2 over 6, the same thing as 1 over 3. In some problems, we're going to use both the multiplication and addition rules. So let's look at this problem from our book, the chance of rolling a 5 and a 4, but in any order. So I can get a 5 first on one die and a 4 on the second die, or I could get a 4 on the first die and a 5 on the second die. I know from my previous slides that the chance of getting a 5 is 1 out of 6, and getting a 4 is 1 out of 6, so that's 1 out of 36. Or I could get a 4 on the first die, and then a 5 on the second die, so that's 1 out of 36 too. And then I'm going to add these two, 1 out of 36 plus 1 out of 36, because either of these cases, the order doesn't matter. Then I would get 2 out of 36, and if I reduce that, that's 1 out of 18. Let's try another question that's not in the book. So let's go back to that couple. What if instead they want to have at least one male child and one female child, and it doesn't matter the order? So they could have a male first and then a female. So that's 1/2 times, remember, 1/2. Or they could have a female first and then a male. That's 1/2 times 1/2. So this is 1/4, and then or I'm going to add 1/4. So that's 2/4 or 1/2. So the chance is half. Another way to solve this problem is noticing that it doesn't matter what the first child is. It doesn't give you a specific order. So the first child could be male or female. It's really the second child that has to be the opposite sex. So that gives you the same answer. It's a half a chance of getting at least one female and one male. These multiplication and addition rules become really helpful when we're dealing with multiple characteristics and traits. For example, I have parents that are shown here, and instead of just looking at flower color or seed color or shape of the seed, we're looking at all three characteristics. It gets really hard to do a giant Punnett square with all of the possible gametes. So I can use probability rules to look at one characteristic at a time. So these are my parents, and I want to look at the possible offspring, this or this. I can look at each characteristic at a time. So let's look at big P, small p from the parents. I know from the Punnett square that the chance of getting big P, small p as a child is half. So that was going to be half. What about big Y, small y from this? If I do a Punnett square, the chance will be half. And then if I look at big R, and this one's small r, and when I see the chance of the child being big R, if I create a Punnett square, again, that would be half. So that's multiplying because I want all three to be true, plus, because this is or, then I want to do these calculations. So the chance of getting big P, small p, looking at just the p's, is again half. If I were to do a Punnett square for just the p's. For the y, big Y, small y, it's the same thing as earlier, so it would be half. And then small r, the chance, if I look at a Punnett square for just the r's, for example, if I were to do that on a Punnett square, that would also be half. So my answer is 1/8 plus 1/8. If I multiply these and add them together, I get 2/8 or 1/4. That's the chance of getting this offspring or this offspring from these two parents. All right, that's the end of our second video. In our third video, we're going to look at pedigrees and patterns of inheritance outside of the normal dominant and recessive outcomes.